Hello and welcome to my video tutorial on how cellular respiration works. So this is a very cool process. It occurs in both plants and animals. This is a common misconception, and it's thought that plants will undergo photosynthesis and animals will, of course, do cellular respiration. However, plants also use their own food to generate their own energy, otherwise how would they grow? So if we were to go to the level of cells, we would notice these small red organelles. As you can see in these diagrams, those are mitochondria, which is where the majority of cellular respiration takes place. So let's start with some context. This here is a person, but it isn't actually. This is a cell. So here we have our cell, or plasma membrane, and in here is a mitochondrion, grossly enlarged. But just for the heck of it, let's enlarge it a little bit more. So we have our outer membrane, our inner membrane, and our intermembrane space. And these are all very important elements to the mitochondrion, as well as something known as the matrix, which is inside of both of these membranes. Now, within the cytoplasm, which is inside the cell but outside of the mitochondrion, the location of this smiley face here, we have something known as glycolysis that occurs. So if we break down the word, we have glyco, which is an older spelling of the word glucose, and lysis, which means to break down. Hey, we just broke down the word. That's a pun. Alright, so glycolysis starts off with a molecule of glucose, which is a 6-carbon chain. Other forms of glucose also are in a ring formation, but for the sake of this video, as the chemical orientation of sugar doesn't completely matter, we'll just stick with the chain. So let's just go with bubbling them off into carbons, because carbon molecules in this case are the most important. And we also have another very important chemical known as adenosine triphosphate. Now, adenosine triphosphate, otherwise known as ATP, is an incredibly important molecule. One of the most important molecules found within animals and plants, or life in general. Water's pretty big too, but this one's huge. So, to look at it chemically, we have a compound known as ribose, another compound known as adenine, and these are attached. There's a few more parts of ribose. And we have 3 phosphate groups. Now, this here is the most important bond within this molecule, because when it is broken, the energy released from the hydrogen bonds between, but the energy released between the bonds between this phosphate that broke off and the water molecules surrounding it releases energy, and that's how a cell does work. So this compound you see before you is adenosine diphosphate, di meaning 2, and there are only 2 phosphates groups. So we can also bubble this group off, and here we have adenosine diphosphate, and if we add another phosphate on, is the adenosine triphosphate. So let's go back to our glucose here. We have 2 of our ATP, which come in, pass on a phosphate, and this creates a very unstable molecule, which proceeds to break down into 2 3-carbon sugars. Now what happens next is we have a compound known as NAD that comes. But stop. What is NAD? Well, this is known as nicotinamide adenine dinucleotide. And a quick chemical composition, we have a ribose, oh that's spelled wrong, adenine, a phosphate, and a phosphate, which looks very similar to ADP, but there are a few more compounds attached. This is another ribose, and we have a nicotinamide attached to the other ribose group. And if we look, there's a quick bubbling of the different compounds that are chemically bonded. You can see a little ADP there. But we have, if you look closely, a little positive there. And if you look on the 2 phosphate groups on the left, you'll see 2 of the oxygens are negative as well. So this shows an imbalance of charges. So we can also add another hydrogen there, and this is going to cause our NAD to turn into NADH, which is a much higher energy compound. And this higher energy will be very important later on in cellular respiration. But if we quickly bubble it off, we can have our NAD plus and our NADH. So these are our compounds so far. Our NADH, our ATP, our ADP, those two are very similar, and our glucose. So 6 carbons. Our ATP comes along, adds a phosphate, splits apart, and now we have our NAD which comes along, which bubble formation, but really it only matters is NAD+. And this comes along with another with an ADP, which another ADP as well, and 2 phosphates, which is just that one part that came off of our ATP. Now these all combine together, so our 2 ADP, our 3 carbon sugar, and our NAD plus with a phosphate. It's a rather complicated reaction. But regardless, we get something known as pyruvic acid or pyruvate, and the phosphate that came off of the 3 carbon sugar combines with an ADP to form ATP. Our NAD becomes NADH, and our phosphates attach to the other ADP, which forms ATP. So in summary, we have 4 ATP are produced, 2 pyruvate or pyruvic acid, and 2 NADH. This is our output. But our input, remember, was our 1 glucose, our 2 ATP, and our 2 ADP and their corresponding phosphates. However, and our NAD+. But however, mid-glycolysis, remember, these ATP broke up into our ADP and our phosphates. So we could really place that in with there, so make it 4, so we don't have to worry about whether or not this ATP actually split off and rejoined with its corresponding chemical. We'll just write it with that, and then 2 ADP also were produced. But regardless, we can just throw that up into our summary corner and move on to our next step. So our cytoplasm is where glycolysis occurs, and also where the entire portion of anaerobic respiration happens, but that will not be covered in this video as I did not draw the slides for it. But anyway, we have within our organelle known as the mitochondrion, the mitochondrial matrix. And so this is where our next process is going to occur. And this one is known as pyruvate oxidation. If we look at this word carefully, pyruvate is our 3 carbon sugar we produced in glycolysis, and oxidation refers to a loss of electrons, whereas reduction refers to a gain in electrons. May seem a little counterintuitive, but that's the way it is for now. So we have our pyruvate that is produced, our NAD plus comes along, and coenzyme A. But stop again. What is our coenzyme A, or CoA? Well, it is a coenzyme, which is very complicated, as you will soon see, but it is also very important. So we have our ribulose, phosphate, adenine, another phosphate, and look at that, we have our ADP with a phosphate attached to the bottom. It's a very interesting compound that occurs all throughout biology, which I'm pretty sure has to do with the polarity, but very cool nonetheless. And then we have some very cool compounds. This is actually vitamin B5, or pantothenic acid. And then we have something known as cysteamine. But regardless, we can just bubble it up and ultimately turn it into a little CoA bubble, because that's all that really matters for an overview of cellular respiration. So pyruvate, we have our NAD+, our coenzyme A, and these all react together and form a carbon dioxide, an NADH, which is a higher energy form of NAD+, and something known as acetyl-CoA. But if we're going to look quickly, we had 3 carbons in our pyruvate molecule and only 2 in our acetyl-CoA. So what happened? Well, one was produced in our carbon dioxide. Pyruvate here's a quick little chemical diagram, does have carbon and oxygen. And 2 of our oxygen and 1 of our carbons move over into our carbon dioxide, and the rest from the 2-carbon chain attached to our coenzyme A. So, as our input, we had our NAD plus and pyruvate and our coenzyme A. And we produced a carbon dioxide, NADH, and acetyl-CoA. But before we stick it into the summary corner, we have to look at something. We produced 2 pyruvates from glycolysis, so therefore, everything must be multiplied by 2, because this whole process occurred twice for every 1 glucose that was produced. So if we have our 2's up there, we can safely stick it up into our summary corner, which is beginning to grow. So, how much energy do we have so far? Well, energy, in terms of a cell, can be measured in how much ATP we have, because ATP is the usable energy form in the cell by breaking off that last phosphate. So this is our useful energy. Well, here we put in 2 ATP, so that's kind of negative 2 ATP, which is our input, and then our output was 2 ATP and another 2, so 4, and then nothing produced in pyruvate oxidation. So we have a total net of 2 ATP produced. This is not actually very much energy for a whole glucose, so we'll see where we get the next amount. So, still within our mitochondrial matrix, we have something known as the Krebs cycle, or the citric acid cycle. There shouldn't actually be an apostrophe there, because a man named Hans Adolf Krebs discovered this cycle. And citric acid is one of the compounds formed in this cycle. It is actually the first compound. And cycle refers to the fact that the final product in this cycle reacts with something to form the first compound in the cycle. Very useful recycling method in cells and life. So we have our acetyl-CoA and something known as oxaloacetic acid, which looks like that if you want to see it quickly. One thing to point out is there are 4 carbons, so this is a 4-carbon molecule. And this reacts with our acetyl-CoA to form something known as citric acid, which, if we look, has 6 carbons. So you can see where the carbons have moved from. This is more of a carbon overview because it's a pretty important concept. Then we go through an enzymatic reaction. All the reactions here are done with enzymes, so this is that the molecules react, but the enzymes never actually change. So we look here, and we have another 6-carbon compound. Then we form something known as ketoglutarate. It's a little blurry, the picture, but this is what it is. But it's only 5 carbons. So what happened here? Well, 1 carbon was actually broken off of this chain and formed CO2, or carbon dioxide. And we also had an NAD plus react and become NADH, which is higher energy, but still unusable by a cell. Then we have something known as succinyl-CoA, which has a coenzyme A attached to it, and only 4 carbons. So you can probably guess that another carbon dioxide was released from this process. So if you look, our 2 carbons from our acetyl-CoA have already been released from the Krebs cycle. So the energy from our acetyl-CoA is being well used in this cycle. And here we go again. Another NAD plus is oxidized, into an NADH. And then we have something known as, I believe it's succinate. Regardless, it is it's between our final steps. And we have here the phosphorylation of our ADP and our inorganic phosphate into an ATP, as well as a coenzyme A is released. But that's not particularly important, and we will see why later. And then we have another 4-carbon molecule formed known as fumarate, and something known as FAD+, which reacts and forms FADH2. But what is FAD or FAD? Well, go over that later because I have a beautiful cycle about to be completed. Then we have malate formed. Not much happens during this step, but the chemical composition changes. And then we have our final reaction back into oxaloacetic acid, in which another NAD plus is formed into an NADH. So here is our Krebs cycle, or our citric acid cycle. In summary, we have on the input side, an acetyl-CoA, 3 NAD plus, and 1 ADP and inorganic phosphate, as well as an FAD plus. And our output, we have 3 NADHs, 2 CO2, or carbon dioxide, 1 ATP, and an FADH2. Now if we look at the rest, the oxaloacetic acid simply reacts back into a citric acid, so there's not much produced there. And our coenzyme A, both between the steps at the bottom and from our acetyl-CoA at the top, this all is part of a large cycle, so isn't really part of an input-output from our glucose, so we can ignore that. So we have, remember, 2 acetyl-CoA produced, similar to our 2-pyruvate. So we are going to multiply everything by 2 again. So we have 2 acetyl-CoA, 6 NAD+, etc. And we can chuck that up into our summary corner, which is expanding across. So what is FADH and FADH2? Well, whoa, it is bright outside, but that's alright. So FAD is known as flavin adenine dinucleotide. So this is very similar to NAD and NADH, but it isn't quite as high energy. And our H2 is the higher energy state, which is a gain of protons and electrons, otherwise known as oxidation. So we have a ribose, an adenine, a phosphate, another phosphate. Hey, look at that, it's ADP. But we have something known as ribitol and flavin. And these two molecules here actually form vitamin B2, each of vitamins. So if we bubble all this up, well, Ri is similar to ribose, so is RAB. But we can ultimately just write that as a B2 and really write that as an FAD for the purpose of this video, but I think it's just pretty fun to see how the chemicals work. So where does the H2 come from? Well, within our vitamin B2, within the flavin, we have, by these two nitrogens, hydrogens will bond to them and form a higher energy state. And this higher energy, like I've mentioned multiple times now, will become very useful later on in cellular respiration. So now what? Well, we've produced quite a bit of FADH2 and quite a bit of NADH, but not that much ATP. So here's what we have so far at the end of this step. But if we look, carbon dioxide is just a waste. It's just breathed out of our system. And then we have our ATP, but that is actually a final product, so we don't really need to use it as input anymore. So ultimately what we have is FADH2 and NADH. So let's go back to our other steps and see what output we had. Well, from our pyruvate oxidation, 2 NADH were formed, so we can make that an 8. And then from glycolysis, we had another 2. So ultimately, our 2 FADH2 and our 10 NADH are our molecules that we need to focus on to now. So within the mitochondrial matrix, as well as the inner membrane, this is a process that occurs mostly in the inner membrane, but goes between the intermembrane space in another cycle form, which is a great way of reusing particles and stuff like that. But let's get on. We have something known as the electron transport chain, which is our final step within cellular respiration. So electrons, of course, are a fundamental particle of the atom. And our transport chain, it involves passing these electrons through a chain of particles and proteins which release the energy of these high energy electrons. So here we have a rather rudimentary diagram of the inner membrane, but let's draw a bunch of phosphate heads and lipid tails. So this forms our phospholipid bilayer, which is pretty self-explanatory. We have our phosphates, our lipids, and two of these occur to make a very interesting layer that occurs in many different parts of life. So here we have a protein, but first, below is our matrix, so in the very center of our mitochondrion, and above will be our intermembrane space, just to get a little bit of context here. So an NADH will come, or this could be an FADH2, but for the sake of describing, we'll just use NADH. And it breaks off and forms NAD plus and a hydrogen proton. And then an electron is released into the chain, and let's draw a quick chain there. So this electron is now passed through these different proteins and these different particles, and its energy is actually released. So how exactly? Well, we have a very large number of hydrogen protons within the matrix, and as the electron passes through the chain, its energy is released and begins to actively transport hydrogen protons across this membrane. And this creates an electrical gradient because, of course, hydrogen protons are simply a proton and have a positive charge. And so this creates a buildup of energy within the mitochondrion. So what do? Well, we have this thing here. This is known as our CF1 particle, or ATP synthase. And these hydrogen protons can only get back into the matrix through this particle. So, as the hydrogen protons move back through, they transfer their energy into an ADP and an inorganic phosphate. And these two here react and form an ATP. But if our hydrogen proton were to just be released back into the matrix, we wouldn't have much of a stream here. It would just, the gradient wouldn't be so equal, and, well, we couldn't produce as much ATP. So we need something that's going to grab our hydrogen protons, as well as this little electron over here, which would get stuck without an acceptor. So we must find a final electron acceptor. Now this can't be hydrogen, but we have something known as oxygen. A circle doesn't really do it justice, but that is oxygen. There we go. A 2 and a G. Much better. So now our oxygen and our electron can now bond with our hydrogen and form the well-known H2O, which often comes out of our bodies, at least in sweat, spit, our breath. So, very cool. So this is our waste product. So as a quick summary, we'll use FADH2 this time. Splits off. We have our FAD plus as our waste, hydrogen protons, electrons. Electrons move through the chain, pumping hydrogen protons across the membrane, which then drive the process of forming ADP into ATP. This is also known as chemiosmosis. Chemi refers to chemicals, in a sense, and in this case ions, and osmosis, of course, is movement across a selectively permeable membrane. So this is just the movement of these hydrogen protons across such a membrane. And so we have our ADP and our phosphate, which form ATP, and then our oxygen comes along and forms an H2O, which leaves as waste. Now, let's do a quick summary. We have our 2-FADH2 and 10-NADH, which are our inputs, and our oxygen, which grabs onto our electron and our hydrogen protons that come out of the ATP synthase, and an ADP and an inorganic phosphate. And our output, of course, is our ATP, which we are trying to produce, as well as some waste products like FAD+, NAD+, and water. But how much ATP have we produced? Because producing one measly ATP after this big process doesn't seem quite worth it. Well, for every NADH, there's enough energy to produce 3 ATP through this process. So, and this process is known as phosphorylation, which is just simply adding a phosphate onto the ADP molecule to form ATP. And our FADH2 will only produce produce 2 ATP, but that is still significant in its own way. So if we apply that to this input-output chart, we have 10 NADH, multiplied by 3, we have 30 ATP produced. And our 2 FADH, multiplied by 2, of course, is an additional 4. So we have 34 ATP produced within the electron transport chain, as well as our waste products of 2 FAD plus, 10 NAD plus. So let's just get some room here and stick this up into our summary corner, which has grown a decent amount. So if we look over to the top left, we can see our NADHs, so we'll add a little +4 there, along with our oxygen and our starting products through the electron transport chain produce all of our ATP. So as you can tell, our summary corner has turned into something more of a summary wall, which is pretty cool. So if we look back at cellular respiration, what it is the breaking down of a sugar, or in this case glucose, into ATP. Now, what we want to do is look at our summary wall and find all the instances of glucose and our ATP production. So, if you look on the very left-hand side, we have 1 glucose produced, and through glycolysis, a net of 2 ATP. Because remember, 2 are lost, but 4 are produced. So 2 are generated. Pyruvate oxidation, not much is produced at all, ATP-wise. And then the Krebs cycle is able to produce 2 ATP. And our electron transport chain is able to form a whopping 34 ATP. So, if we write all this out, we have a glucose is transformed into 38 ATP, which is a very impressive figure, but really it's just enough for us to get by in our daily lives. An interesting fact, though, our bodies are able to produce as much ATP as our body weight. So that just kind of shows you how much ATP is produced if every day we produce our body weight in ATP, which of course is just simply breaking and reattaching a phosphate. So this here is our cellular respiration, which, very cool process. And thank you for watching this video. And yeah, hopefully I'll be back with more. See ya.